Hi everyone, we are here to present on vaccination of COVID-22X. I'm Acquah Emmanuel, with me is Chaeyeon Kim. Vaccination is the administration of agent-specific but safe antigenic component that in vaccinated individuals can induce protective immunity against the corresponding infectious agent, such as viruses. Vaccines tend to control viral infection and pandemics, strengthening training immunity across history. Vaccine is one of the world's greatest inventions in medicine by preventing 2 to 3 million deaths each year. What do we know about vaccines? Vaccines come in different forms, such as live attenuated vaccine, live viruses, and inactivated vaccine. These tend to build protection as well as boost immunity, but sometimes they can also develop or induce autoimmune responses, leading to autoimmune diseases. Let's look at how vaccines are being developed. There are 6 different stages in which vaccine development takes place. The first is the exploratory stage, whereby the genetic makeup of the virus are being identified. Next is the preclinical stage, which includes animal testing of these vaccines that are being generated. Once it is done and successful, the clinical trial then begins. This involves 3 different phases involving different number of participants. Next, once it is successful, the regulatory review or approval of this vaccine is being done, leading to the mass production of this vaccine, which then moves to the quality control and the distribution. What do we know about COVID-19 vaccine? COVID-19 or the SARS-CoV-1, MERS-CoV, as well as SARS-CoV-2 are beta coronaviruses that have one specific feature, that is the spike glycoproteins. These have receptor binding domain that bind to the cell surfaces of the human. And the receptor that these viruses bind to include the ACE2. So most vaccines tend to target this receptor binding domain, and therefore most viruses vaccines are being generated through this antigen for COVID-19 vaccine. What do we know about COVID-19 vaccine? There are so far about 41 candidates or vaccine candidates being generated through several forms using different vaccine technology, such as mRNA, DNA, protein-based, viral vector, live attenuated technology that are being used, which are under either clinical or the clinical stages. Let's look at some of the challenges related to pandemics in the previous and current pandemic. Some of them include the enhancement of diseases. Some vaccines, when introduced into humans, tend to increase the infection even when compared to unvaccinated people. This can also be a problem to the whole world population. Next is the waning of the antibody response. Sometimes after 2 to 3 years, for example, SARS-CoV-1 and MERS-CoV infections, the antibody protective response is being lowered, and therefore people or individuals can be reinfected again. Next is the long time frame. It takes a longer time for vaccines to be developed from different stages. It takes about 5 to 15 years to come to the market. Also, the funding or investment in vaccines is huge. It takes several billions of dollars, and also in terms of production and facility capacity to develop vaccines. Next is the pandemic-related challenges, which involves the previous ones, which includes the susceptible small animals in large numbers. It's also needed. So far, it is difficult for animals to be infected with this coronavirus, and also therefore it's a challenge. Next is the strong cross-neutralizing antibody to SARS-CoV-2. Therefore, so far there is no cross-neutralization antibody to SARS-CoV-2. And next, since it affects older people, higher neutralization titer for older people is being needed. And also, these beta coronaviruses have conserved epitope with mutation, which tend to create resistance to antibodies that are being used. I would like to end here with my presentation and hand over to my partner to continue. Thank you.
Issues and suggestions for upcoming pandemic virus vaccine can be represented to time and expense costs, and its distribution. Not only for coronavirus, general vaccine development pipeline has low success rate but high financial risk, making the companies fear to invest time and expense costs in vaccine development. The prediction of candidate selection is essential for reducing time and expenses, enabling the lower risk on vaccine development and fast-track pandemic paradigm. However, the COVID-19 vaccine is expected to develop in 2 years, and the upcoming vaccines are predicted to develop in even shorter term, thanks to fast-tracked pandemic paradigm. And also the prediction of period of epidemic disease emergency using AI, such as blue dot outbreak risk software, can play for time. Here suggests the pandemic paradigm versus classical procedure that Acquah explained before in COVID-19 vaccine development. In classical procedure, the vaccine distribution is expected to start at May 2036. However, when we suggest the pandemic paradigm, the shortened development time, such as start preclinical trials early and receive academic research period, can advance the date to April 2032. And clinical trials can be overlapped and compact by allowing the large-scale tests sooner, and also approval and distribution stages can be skipped as an emergency provision. Distribution and mass production part are the most preparable part as preparing for the factorizing pipeline. So in conclusion, the fast-track procedure allows the emergency vaccine produced by February 2021, which is very shorter term in development of COVID-19 vaccine. The prediction of vaccine structure can help solving the issues, especially the coronavirus has similar spike proteins, and the genetic data is openly provided. COVID-19 has 80% similarity with SARS virus, and both vaccines target ACE2 receptor, as the right figure suggests. So for example, coronavirus spike proteins has been simulated in RNN training using previous coronavirus genetic codes, as the left figure from the Lisa C. Crossman paper suggests. The distribution of vaccines and at low cost and the mass production, which is the last issues we will address, can be enhanced with a better delivery system and producible vaccine approach with immunogenicity boosting strategies. For example, the microscopic needle, which deliver antigen by dissolving microneedle arrays, are cost-effective and easier to manufacture. Economic evaluation of microneedles are sharing more than 20% to be cost-effective. So, but there still exist the remaining issues, which the coronavirus has its viral variants. COVID-19 has been reported that the mutated spike protein G614 has been displaced the original protein G614, as the below figure shows. The G614 is the blue one, and the G614 is the orange one. So the variable in epidemic virus may disturb the vaccine development, and further studies have to trace the virus after its appearance. Thanks for the listening, and this is the reference that we cited.